So yesterday, basically, we talked about the intro to hematology. We talked about the different cells, those kind of things, and Monday also. So today, let's talk about hematopoiesis. Okay, that's a big term. It's a new term you probably have never seen before in your life until today, because it's a specialty term. I know you guys may have had biology, maybe even had anatomy, but I'm willing to bet you have never seen this word before. Okay. So basically, anytime we see the word poiesis, the part, I guess this would be what, the suffix, poiesis in English, it means to form or to build, okay? So hematopoiesis simply means to form the blood cells, and that's what we're going to talk about for the next couple of chapters. This chapter is chapter 3 of your book, so if you got it, we'll be in chapter 3. Next chapter is chapter 4, and then we'll have a test. Okay. If you looked at, I kind of reviewed what I've got set up. I kept mentioning the units. So unit 2 is basically the erythrocyte, hemoglobin, and the CBC is what we're going to talk about. So it's like chapter 5, chapter 6, and chapter 10, I believe, is what they'll have. So unit 2 will be a test also, okay? And then unit 3 is the RBC abnormalities. So things like anemias, hemoglobinopathies. And then unit 4 is leukemias. So things with white blood cells, the abnormalities of white blood cells. And then chapter 5 is hemostasis, which is the clotting stuff. And then chapter 6 or unit 6 is body fluids, okay? So that kind of sums it up, and I got that on your syllabus somewhere along the way, okay? But if not, you've got it, the PowerPoints already, so you can kind of go through this. All right, so let's talk about hematopoiesis. So hematopoiesis begins about as early as the 18th day after fertilization. So once the fetus is born or conceived in the womb, you have to have red cells to basically carry oxygen. That's what they do. There is no other means in our body to get oxygen to tissue and carbon dioxide away from tissue except the red blood cell. Okay? Some carbon dioxide is actually dissolved in the plasma, but very little carbon dioxide. So the only way that you can get gas exchange to occur for your tissue is through red blood cells. Okay? So the very first cells that are formed in the fetus is, or in the embryo, is the red blood cells. Okay? So, during fetal birth, or fetal development, of course, you got your uterus, okay? Uterus is kind of pear-shaped organ, okay? So you got basically your placenta. The placenta attaches to the uterine wall, okay? Just like this. Then there is a little area of the placenta that is known as the yolk sac. Okay? The yolk sac. And this yolk sac is actually where hematopoiesis starts. So it's actually extra embryonic. So anytime you see the word extra, it means outside of. So extra embryonic development of the red blood cells. They make a few white blood cells. They're kind of similar to monocytes that we talked about yesterday. They just refer to them as macrophages, though, because they're extra embryonic. Okay, so they're not in the peripheral blood yet. Okay, because technically at this point, at 18 days, there are no vessels. Okay, it's just cells. Okay, but those cells are alive. So therefore, they have to have oxygen and CO2 exchange. Okay? So red cells do take care of this at about the 18th day. Okay? Now, as the cells or the fetus or embryo develops further, then the liver begins to take over. Okay? Now here's a chart. Now this is a mammal. I think this is a rat or something. But mammalian development is always the same for every animal, okay? I know we don't like to talk about that, but we are an animal, okay? Matter of fact, we have a tail at a certain point in our development as a fetus, okay? But our DNA is designed so that we don't have a tail, okay? Sometimes rare things happen, and there's actually been kids born with tails, okay? Because they didn't develop like they were supposed to, all right? But we do, okay? The point is, the yolk sac basically is part of the placenta until a certain point, okay? So once the yolk sac takes over, okay, so you can see from days, all right, as you go through, then after a few days of gestation, there is an area around the aorta, okay, right here. This will become the aorta of the heart and the heart itself. It's known as the aorta gonad mesonephrins, okay? So what's going to happen is this area is eventually going to turn into the aorta in the heart. It's going to turn into the gonads, so like male testicles, female ovaries, and then you're going to have these primitive kidneys, okay? Nephros is always kidneys, okay? So this area, the AGM, picks up hematopoiesis the next level, okay, as the baby is maturing. Now at this level, we still have red cells, and then we start actually making some primitive white cells, okay? They're very similar to lymphocytes, but not quite. So once the AGM takes over and begins to produce hematopoiesis, then you get the hematopoietic stem cell, okay? HSCs. Okay? It stands for hematopoietic stem cell. Okay? Now, we're going to talk a lot about this guy in the next few chapters. Okay? One thing I need to make sure you guys understand. There is a difference between all the stem cells. Okay? We have stem cells that have specific jobs to do. Alright? In this case, we're talking about stem cells that make blood. Alright? That's the reason they're referred to as hematopoietic stem cells. Okay? But we also have stem cells that make nerves. We have stem cells that make muscle. We have stem cells that do make our organs. Those kind of things. Does that make sense? So when we start talking about stem cells, I don't want you to just think, well, there's only one type of stem cell in the body and it does everything. Okay? That's not true. Alright? There are different populations of stem cells. Okay? But what we're going to discuss the most is the ones about hematopoiesis. Okay? Because that's what we're dealing with in hematology. Okay? Does that make sense, guys? Alright? So, during this time, the yolk sac begins hematopoiesis. It's extra embryonic. Okay, so it's outside of the embryo. It occurs. And then, of course, it goes in through the vessels to get to the embryo. All right? Now, the AGM begins taking over hematopoiesis also. Okay? At this point, we're making red cells. We're making some white blood cells. These kind of things. All right? Now, as time goes on, and I want you to pay attention to this, because as the days go of gestation of the baby, you can see that there's overlapping, okay? There's never a time where one stops and then the other one starts, okay? They start working in unison with each other. That way we're never without cells. Does that make sense? We can't be without cells because the more we grow in the embryo and the fetus development, the more oxygen, the more CO2 is going to be created. Does that make sense? So we've got to continue production. All right? So eventually, if you'll notice, it goes yolk sac, AGM. The placenta plays a little bit of a role, so this would also be a form of extra embryonic. But then eventually what's going to take over? The liver. Okay? The fetal liver is very, very small, but it begins to do that. All right? So do adults. Adults, we also still have a little bit of hematopoiesis that takes place in our liver. Okay? So that never stops as we grow. Does that make sense? Okay? But once the liver begins doing it, the next tissue is the thymus gland. Okay? Thymus gland, I talked a little bit about it yesterday. We're going to talk a little bit more about it today. But it's the area where lymphocytes are matured. Okay? Now, lymphocytes are formed in the bone marrow. So when in gestation, or while the baby is developing in the uterus, these are really, really primitive lymphocytes. Okay? Because the bone marrow is eventually going to take over and start making them, and then it continues to do that job throughout adulthood, okay, until death. All right? But as we go, all right? So one thing that's not mentioned in the embryonic form of hematopoiesis is the spleen, okay? But the spleen does play a role, even though it's not made mention of on this chart. Okay, the spleen does have in the embryo as it does in the adult. And those are the things we're going to talk about today. We're going to talk about the bone marrow, we're going to talk about the liver, and we're going to talk about the spleen and the thymus glands. Okay, those are the four major hematopoietic tissues that we're going to talk about. Okay, does that make sense? All right. So from 18 days of gestation, we have to start making red cells, okay? The reason that we don't have to before 18 days is because it's just a ball of cells, and it's not. By the 18th day, we're starting to put a little structure into the body, or into that embryo, and it's starting to kind of look like an embryo. Does that make sense to you? All right. Any questions about that at this point, guys? Okay. All right. So the thymus is the primary site of the T cell production. We talked a little bit about T cells yesterday. What are T cells? From lab yesterday. What classification are T cells? You remember? Lymphocytes. That's exactly right. And I told you there's two main ones. What are they? Y'all remember? T4 and T8. There you go. T cells. Now you were right. Somebody said B cell. Yes, those are the two types of lymphocytes. You're exactly right. But in T cells, there were two types that we talked about. And then there's a natural killer cell. But it's not referred to as a T cell. It's actually just called a natural killer cell. Okay? But T cells are T4, T8. All right? So these are the ones that develop even at the embryonic stage and in fetal development. And then, of course, they remain active up through the neonatal period into childhood. Usually by the time they're about 15 or 16, the thymus gland begins to atroph, okay? So that means it begins to shrink. And then by the time we're my age, I don't have a thymus, or I have a few cells, but not enough to thymus that you could even detect it on a scan, okay, as you go, all right? And so this is starting that immunity. Remember, we talked about the T cells are cell-mediated, B cells are humoral immunity, okay? So even at the fetal development, it still has to have some immunity, okay? Because mom can expose him to lots of things, or it to lots of things, okay? All right, questions about that, guys?
Say it again. T cells are
their immunity. Play the role in our immunity. It's cell-mediated immunity.
The
Okay.
T4 and the T8.
Yes. Okay. And so it has to have them because it still can be infected. It can still have foreign bodies come into it, even though it is in development of the womb. Okay. All right. So first development T cells use a different set of genes, and this is one thing that's really, really unusual. Because at the fetal level, it has a whole set of genes that only come on while it's in the embryo. Does that make sense? Okay. So if you don't understand that, all right. So what is it? BR. Is it BRC1? Maybe it's a BCR1. I think that's it. BCR1, is that breast cancer gene? Is that it?
Yeah.
What is it?
BRCA.
Yeah, is it?
BRCA.
BRCA. Okay, I knew it was BRC something. I'm just pulling this off the top of my head. Okay. So we know that there's a gene for breast cancer. It's actually a couple of genes. We know that give you an increased chance if you have those genes to having breast cancer, right? Does that mean if you have the gene, you're going to get breast cancer? Huh? No, it doesn't. It gives you a higher percentage, but what has to happen for you to get breast cancer? Gene has to be what? Cut on, right? In other words, that gene has to be activated and cause a protein or whatever the mutation is to go into your body before you actually get breast cancer, okay? So that's what we're talking about in fetal development of white blood cells. They actually have a certain set of genes that's different than us as adults that helps them to make T cells. And then once they reach a certain age and are born, then that gene gets cut off. And your body doesn't make those particular proteins to make those T cells develop like that anymore. And it turns on your adult side. And then you start making T cells like me and you do. Does that make sense? Okay? So that's kind of weird. But that's how genetics work. All right? So these basically, all right, so let me stop right there. So do y'all know what a receptor is, science-wise? Have you ever seen this term before? Okay. What's a receptor? Tell me what that means.
Structure that receives.
That's it. That's exactly what it is. It's a structure that receives things. And so, like, we're going to talk about this later on, but we've got lymphocytes, okay? And these lymphocytes have certain structures on their outsides, okay? Okay? And they're all different shapes because they all play a different role. And so they're all different in the cytoplasm or in the cell membrane of these cells, okay? So we have a cool cell called a dendrite cell or a dendritic cell, however how you want to say it. He's a narc. And what an narc is? It's a tattletale. That's what he does. He actually goes around your body all the time and tells on bacteria, or tells on, not bacteria, but tells on viruses, tells on foreign animals. And what he does, not only does he tell on people, and he looks very similar to a lymphocyte, so it's kind of hard to tell the difference in them. And he grows up, guess where? In the thymus, just like lymphocytes do. Okay? So what he does is like, let's say our COVID virus. Our COVID virus, our dendritic cell comes up close to that virus and it says, oh, hey, you're not supposed to be here. So what he does is actually, he kind of takes a picture chemically of that virus. And I know it sounds crazy, but that's exactly what he does. He takes basically a chemical, almost a chemical sample or picture of it, and then he runs back over here to this T cell and says, hey dude, there's this foreign dude over here, and this is what he looks like. You need to go get him. And he goes and gets him. That T cell or that T4 or T8 cell, whichever one it is. Okay? Alright? So receptors are really, really cool. And us as adults, we have a particular set of receptors on all our T cells. Okay? And what the thing's talking about is fetals have different ones than what we have. A whole total set. Because their immunity is not as, I don't want to say as good, it's not as necessary because they have mom to take care of them. They're in an enclosed area that's like a vacuum. So in theory, nothing should ever get to the fetus. the embryo. Does that make sense? So they have a very weak immune system and a very different immune system because they're not being exposed to everything like me and you are. Does that make sense? Okay, awesome. All right. So lymph nodes and spleen. So we know that embryos also have lymph nodes and they also have the spleen. Now in the lymph nodes and spleens of the embryo, this is where B cell differentiation is occurring. We talked about B cells yesterday. B cells, of course, are a type of lymphocyte also. The difference is B cells are usually larger lymphocytes, and then they mature into another cell. And this cell is a weird looking cell. It is, the nucleus is usually perfectly round. It's called a plasma cell. Alright? Plasma cells are usually purple cytoplasm. The nucleus is eccentric. What's eccentric mean? Anybody know what eccentric means? Kind of weird, right? Eccentric means it's kind of different or not the same, kind of shifts to the left or shifts to the right. That's what eccentric means. So in this case, most of the time your nucleus is eccentric, so it's usually pushed to one side. And what's really cool about it is it actually has a lot of endoplasmic reticulum, which is because it makes a lot of proteins. That's what the cell does, its job. And so you get what we call a doning effect. And I'll show you guys some plasma cells a little bit later on. But then that doning doesn't stain. It doesn't take up the right stain. So you get a white area around the nucleus. And you know how at the beginning of the day it's dark, and in the morning, you guys ever seen the sun come up? Okay? So as it's dark, but then as the sun starts coming up, what do you get right between the horizon and the sky? You get light, right? It starts turning white. That's called a doning effect. Okay? So the doning effect is one of the things that gives away plasma cells. Okay? Plasma cells then mature into what's those? Not science, people. What are those? That girl. Antibodies. Small. These are antibodies. Okay? And they can be IgG, they can be IgA, IgE, IgM, GA, EM, and D. Even though we don't know what D does. D has no function, as far as we know. We do produce them, but we don't know what they do. Nobody knows what they do. But it can be any of those. But every one of them are going to come from a B cell. A B lymphocyte is going to mature into a plasma cell when it's stimulated, and then it's going to become antibodies eventually. Okay? So that's how it works. So when our spleen, for embryos, the spleen and the thymus, or I'm sorry, the lymph nodes in the spleen, are the site for late B cell. Now in our adult body, though, it is the lymph nodes and the thymus is where they stay, and mostly the lymph nodes. Now we do send a few B cells back to the bone marrow just to kind of help us, for protection, that humoral protection. So you will see a few B cells, and you may even see a plasma cell on a bone marrow aspiration. Now, was Candace here yesterday in lab? So she sat by you. Did somebody sit by you yesterday, at least? I didn't think so. I thought she was out too. All right. Sorry, guys. I have ADD. Start thinking about something, I have to stop and write it down. If I don't, I'll forget. All right. So any questions about embryonic hematopoiesis? Okay. Very, very quick overview. All right. So let's talk about adult hematopoietic tissue. Okay. So adult system, like we said before, includes the bone marrow, thymus, spleen, and the lymph nodes. Okay. Those are the main ones. The liver can do a really not very good, so not efficient form of hematopoiesis, but we don't talk about it in the adulthood, okay? Because it doesn't do that good of a job. But your bone marrow, of course, is your main one. The thymus, because that's where your T cells and B cells, your lymphocytes mature. And then your spleen, we talk about hematopoiesis because hematopoiesis doesn't just mean the formation, but it's the lifespan of it. So from birth to death is what hematopoiesis really is. Okay? So your spleen plays a huge role in destroying cells also. Okay? And we'll talk about that in just a few minutes. All right? Questions? Anybody? Going once, going twice. All right, so let's talk about the bone marrow. So the bone marrow is very, very important to us. Without bone marrow, you will die. Okay? Does everybody understand that concept? If you don't have bone marrow, you will die because your bone marrow is what produces those cells, especially the red cells, that what keeps you alive. Okay? You have to have that to survive. All right, so I'm fixing to have to figure out something real quick here, because I'm going to show you guys a video if I can make these speakers work. And don't know that I'm going to be able to make these speakers work because these speakers are old as I am. Ha ha! Got you, sucker. Maybe. Got you. Whoo, got power. All right. So, external speakers. Medium. Okay. All right. So we're going to talk about the bone marrow in general. So your bone marrow. All right. Bone marrow is the site of cell development for myeloid cells. Okay. Myeloid cells. This is going to be your first term for this. Myeloid are eos, neutrophils, basophils. Okay. All right. Erythroids are red blood cells. Megakaryocytic are your platelets, thrombocytes, and then early stage of your lymphoid cells. Okay? Now, it's going to get a little confusing in the next chapter, because in the next chapter we're going to see this big flow chart of cells. Okay? All right? But this is where all four of your stages are actually produced, but they don't necessarily stay there. Where they talking about mostly the lymphoids, okay? They're formed in the bone marrow, but they leave for maturation to go to the thymus and the lymph. Yes?
Platelets, just
Yes,
platelets? What
yes.
was the next one?
That's it.
Okay.
Lymphoid, so lymphocytes.
Okay.
Okay? All right, your thymus, your spleen, and your lymph nodes are also sites of later lymphoid cell development. Okay? All right, so two terms. There is a term called medullary. I told y'all vocabulary is very, very important. Uh-huh.
What'd you say?
I said I told you guys vocabulary is very, very important, right? Remember me talking about that the very first day? All right, so medullary. Medullary. Okay? Medullary hematopoiesis. Oops. S-I-S. Right? Is that how you spell it? Did I spell it right? Or is it I-E? P-O, is it I-E? And then S-I-S? Is that right? All right. Medullary, if you see medullary hematopoiesis, this is the bone marrow. Okay? That's the term. If we're talking about stuff being formed in the bone marrow, that's what we use. We say medullary hematopoiesis. Okay? Now, if we say extra medullary, and I'm just going to say hemat, so you know, we're talking about hematopoiesis, we're talking about the spleen, thymus, okay, and what's the other one? Lymph nodes. And then I'm going to put in parentheses liver. Okay? But these three are mostly what we're talking about. Okay? So if we start talking about that, it's referred to as extra medullary. Okay? Everybody good with that? Make sense? Excellent. All right. So, bone marrow and the thymus. These are your primary lymphoid tissues, okay? Primary. So what if I say something's primary? What does that mean? First. That's where it's going to occur. Now we can have secondary stuff, but these are the two main ones, okay? So like later on, your lymph nodes would be secondary development, okay? Or secondary lymphoid tissue, all right? Matter of fact, just your lymph nodes, even your tonsils are considered your secondary lymphoid tissue, okay? All right, so T and B cells develop from non-function precursors, okay? As you may know what a precursor is. You know what that term means. What is a precursor? Before. That's exactly right. So, you remember yesterday in class, if you were here, I told you guys there's a myeloblast. We talked about how it's a big cell, high NC ratio, all that kind of stuff. And then I said the next one was a promyelocyte. And then we said the next one was a myelocyte. And then we said at that point we could tell if it's an EO, a baso, or a neutrophil, right? Remember that? And then it talked a little bit. I told you there's a couple more until we get to the band and the seg. Okay? Promyelocyte is the precursor of the myelocyte. Does that make sense? It's the maturation right before the other one. Okay? So when we're talking about T cells and B cells, there is a lymphoblast, a prolymph, or lymphocyte, and then a lymphocyte. Okay? So this and this one are precursors to the lymphocyte. Okay? Whether it's a T cell or B cell, B lymphocyte. Okay? Does that make sense? All right? Now, listen to me. This one and even this one, in theory, you can't look at it and tell if it's a T or B, right? Remember I told you you have to use a special technique called flow cytometry to know what they are, okay? Even though these are blasts, if we do flow cytometry, we can tell if they're EO, baso, or neutrophil, or if they're T or B, okay? Even though we might not be able to look at them and tell, chemically or immunoassay wise, we could. Okay? Does that make sense? Alright? Because, we talked about receptors a while ago. We know what receptors are supposed to be on cells. And so we can actually test for those receptors. We don't call them receptors, but we call them antigens, but that's essentially what they are, is receptors. Does that make sense? Okay? Alright, so when we talk about precursors, at this point they're non-functional. Okay? So, my 16-year-old at this point in his life is non-functional. Okay? He doesn't have a job. He goes to school. He doesn't know what he wants to be when he grows up. Does that make sense? Doesn't know where he wants to go to college yet. None of that stuff. He knows he wants to be a billionaire. He don't know how he's going to accomplish that. Does that make sense? So right now he's non-functional. Okay? So all of the precursors that are in the thymus gland, these T cells and B cells, they're non-functioning precursors. Okay? But then they're going to reach an age where they become immunocompetent. Okay? So hopefully by next year, the end of the next year, middle of his senior year, he will decide, oh, I think I want to be a lawyer. Oh, I think I want to be a doctor. Oh, I think I want to be a teacher. Okay? If he does that, he becomes a little bit functional, right? At least he's been exposed to something to make him say, hey, I might want to do that. Does that make sense? So our body's exactly the same way. Our cells get exposed to things that make them capable of going to work. All right? Once they do that, then they're referred to as immunocompetent. That means they have the ability to go do some work when it's necessary. Does that make sense? Okay? And that's what we want our cells to be. We want our cells to be immunocompetent. And this occurs in the bone marrow and also in the thymus. Okay? Once they're formed in the bone marrow, they're basically designed to say, okay, I'm going to become a T cell, I'm going to become a B cell, based off of chemicals. They're called cytokines, if you want to write that word down. They're called cytokines. Cytokine. E. I think there may be an E on it. I think so. It may not. I don't remember. But either way, those are the signals, the chemical signals, that tell that my brain just went blank for a second. The stem cell, the hematopoietic stem cell, that, hey, we need a T cell. Does that make sense? Or we need a B cell, or we need a neutrophil, or we need a basophil, or we need a red cell. Whatever it is that we need, it's always going to be based off of the chemicals that it's exposed to. All right? And so that's important to us as adults. All right? So, spleen and lymph nodes are known as our secondary lymphoid tissue. So this is where immunocompetent cells that were formed in the bone marrow and the thymus, they go to hang out to eventually become competent in their job. In other words, they're ready to do what they need to do. In other words, they become mature, active cells to do what they need to do. Does that make sense? Okay? All right, any questions about that? All right, so let's talk about the bone marrow in general. All right, so that was just a little overview. We're going to talk about each one of these in more detail as we go. All right.
The procedure starts by finding the correct level for the biopsy. The biopsies are taken from the upper iliac crest with access via the ventral spine. The midline of the spinous process is identified and the iliac crest is located by palpation. When the correct point is found, the puncture point is marked using a straw. This is to avoid losing the marking after washing. The area around the puncture point is washed with colored chlorhexidine. The procedure is carried out aseptically, but without sterile drapes. The area around the puncture point must remain sterile during the whole procedure. Correct placement of local anesthesia is important for how the patient experiences the procedure. 10 milliliters of xylocaine with 10 milligrams per milliliter of adrenaline is normally used. When the needle reaches the periosteum, the anesthesia is given time to work before the numbed area is expanded by angling the needle in 4 directions. To avoid unnecessary trauma to the skin from the aspiration needle, a small incision is made in the skin with a scalpel blade. Disposable and reusable aspiration needles are available. A reusable needle is used in this video. The needle is pushed toward the posterior iliac spine and rotated with careful pressure until it goes through the cortex. When the needle is in the spongy tissue, the trocar is removed and a 5 milliliter syringe is connected to the needle. 0.2 to 2 milliliters is quickly aspirated for a regular bone marrow smear. 3 small and 7 larger smears are placed on slides. The slides are tilted and the blood, which collects in the lower edge of the smear, is removed before it coagulates. We then use another slide to make a few regular smears and some which are squeezed.
Called squash. They squash them together to make them.
The smears are dried with a fan before fixation and staining. A new aspiration needle is used for aspiration for flow cytometry. 0.5 milliliters of monoperin is placed in the syringe in advance to stop the aspirate coagulating. 4 to 5 milliliters is aspirated with the syringe. The aspirate is quickly moved to a specimen glass. Finally, a punch biopsy is taken. A disposable needle is used. The needle is inserted in the same puncture wound and pushed toward the posterior crest. The needle is rotated with constant pressure until it penetrates the iliac crest. Care is taken to ensure that the needle does not slide into an area that is not anesthetized. The trocar is removed from the needle. The rotation of the needle is continued until it has penetrated deep enough into the bone marrow. The spoon is inserted into the needle. The biopsy needle is rotated without pressure in both directions to cut the biopsy loose. The needle with spoon and biopsy
Pulling
is
out
pulled
will
out.
be a little core. You ever seen them aged trees? Like an FFA if you was in school?
The spoon is
Push that
carefully
thing all
removed
way
from the
through,
needle,
pull
and the
out.
biopsy is put in
That's what it
fixation
is.
fluid using a blunt trocar.
The trabeculae.
The incision is
we're gonna
closed
talk
with
about in
strips
just a minute.
and covered with an
That's
adhesive
what it looks
bandage.
like. Okay, so this is a bone marrow aspiration. Not fun. It's not anything you really want to have done if you can help it. You know what I mean? I have done several of these
What we have to do?
in my career. Well, you will go to a bone marrow. Hopefully, you should when you do your clinicals. You should. That's one of the things that we would like for you guys to do. It just depends. Like, so nobody. A lot of times hospitals don't do them anymore. A lot of times hospitals will do them in surgery. They'll actually take the patient to surgery. The person usually doing it is a doctor, of course. A lot of hospitals will allow, it's usually a pathologist that does it, but some oncologists do their own. But I have seen general practitioners do these also for their patients. You know, they suspect that their patient has leukemia or something like that. You need to look at a bone marrow aspiration. Okay. All right. So they're usually awake unless it's kids. All right. So any of you get sick watching that? All right. So I've done 3 of them in my life, in my career as a tech. Okay. First 2 was a student. I was a student at Marshall. Okay. I have never made it through but one of them that I didn't have to step out for a minute. Okay. And I feel like I'm a pretty tough guy. Okay. But the first one that I did, I was a student, went in, it was an older guy. They had the room hotter than 3 hells. Nobody was talking. It was just like dead silence in there and everything else. And so we got our tray ready. We got there. He started working. The dude was like moaning and all this other crap. And I had to walk out. I had to step out. I told the guy, doctor, I said, I can go step out for a second, be right back. So I went out, got my composure, went back in, finished up what I was doing. Done my slides and got everything done. So I made it through it. Second one, same way. It was a younger guy. I was about my age. Room was hotter than 3 hells. Again, nobody was talking, whatever. So I had to go out again when we started. Never passed out, but I did get close on that one. And so the third one, I was out of school. I was at Medical Center East, which is St. Vincent's East now. And so, no, I'm not being ugly, I'm just telling you the truth. So this little Chinese girl, little Asian resident that we had, literally she's about this tall. No joke. I mean, I'm not even exaggerating. Real bitty thing, resident on second shift. I was on second shift. Came in and called and said, hey, I need to do a bone marrow. And I said, no, we don't do bone marrows on second shift. I said, you can schedule that on day shift. There's no reason for us to do this. And she said, well, I really need to do this. And probably did need to do it because she probably had to check something off in her, residency competencies. So, but I told her no. I said, no, we're not doing it. I said, you can call tomorrow and set up an appointment. Well, about 30 minutes later, I get a call from our pathologist, who's their doctor of the lab. And he said, Joseph, you need to go do that bone marrow. And I told him, I said, Dr. Davis, I said, no. I said, we're not doing it. I said, there's no reason. I said, you let this one do this, and from here on out, we're going to have to do them on second shift. And I said, I'm not doing them. And he said, Joseph, go do the bone marrow. And I was like, okay, So I got my stuff together. We went up there. All right, so in an adult, okay, so let's go ahead and talk about the bone marrow. In an adult, okay, couple of places. Skull, iliac crest of the pelvis. Okay, so like you little skinny girls with your little skinny jeans that you see hip bone right there, that's your iliac crest. I'm fat. You don't see my hip bones anymore, right? But that little area right there on the crest of the iliac or the ilium, okay, is where we do them. That's the first choice, okay? Second choice is your sternum, okay? You can do the skull or you can do vertebrae, okay? And of course, nobody does vertebrae because your spinal cord's there, right? So you wouldn't ever want to do your vertebrae. And your skull's just not applicable because, you might puncture the brain. So guess where we do it? Iliac crest. That's it. That's where most people do it, okay? That's why we do them there is because those bones still have full red marrow. Okay, so we're going to talk about some things in just a minute, but to get my story done, okay? So I get my tray, all my equipment that I need. I head up to the patient's room. I get there, and this little Asian girl, okay, I'm sitting there. I've got my back turned to her because I'm setting up the tray. That way I can turn and get the samples and then turn back and do what I need to do. Make the slides, put the biopsy in, and all this other stuff. So I'm turning, and I hear her back here behind me talking to the patient and everything, and I'm not paying a whole lot of attention because I just do that, So I got my stuff ready, and I turn back around, and this chick is on this guy like this. She has crawled on top of him and straddled him, and she is going to collect this bone marrow from his sternum. And I was like, what the hell are you doing? And she was like, I'm going to collect this thing. I said, you're not going to use his iliac crest. And she said, no, I'm going to collect it from his sternum. And I said, whatever, you know. So she cracks this dude's chest, you know, sternum, gets her marrow, pulls it all out, hands it to me. Guess what? I ain't passed out this time because there's too much going on for me to even think about passing out. So I get my stuff, I've got my tray, I get all my slides, the biopsy, all that. She's taking the patient, I'm heading out the door. Well, when I get back to the lab, Dr. Davis calls, said, Joseph, did you do that bone marrow? I said, yeah, I did it. And I said, listen, I ain't doing another damn bone marrow, ever, in this hospital, you know? He said, Joseph, why? And I said, you ain't gonna believe what this chick did, you know? And he's like, what? And I said, she collected it from his sternum. And he was like, really? And I was like, yes! I said, I'm never doing another one again. I said, that's traumatizing to me, you know? But anyway, so that was my bone marrow. That was the last bone marrow I ever did. So hopefully you guys will get to do one in class, okay? So if you do one or get to do one in your clinicals, you know, just be aware that you may get a little sick or whatever, and you may have to step out, and that's okay. I don't know, just the thought of them punching that bone, you know, I guess. I don't know.
Does the video make you sick?
No, the video doesn't make me sick.
Okay.
But in real life, it did. You know, I was young, naive, didn't realize what all was going on. I've done an autopsy now. I mean, that don't bother me anymore.
Well, they're dead.
Yeah, that's what I'm saying. But anyway, questions about bone marrow. So I recorded that little video, so watch it again after we talk about this, and you'll kind of see what we're doing here. All right? So, bone marrow. So this is where your blood-forming tissue is found, okay? And it is between the trabeculae, okay? So trabeculae is essentially, all right, so like if you got a matrix of something, okay, bone, whatever, this areas, like the open areas, are known as your trabeculae, okay? And there's pictures in your book, and I think there's a picture on the next couple of slides, okay? The trabeculae are just little open areas of your bone, all right? This is where the red marrow forms, all right? And I keep saying red marrow because there are two different types of marrow. There's red marrow, which is what we want to make the cells, and then there's yellow marrow. Yellow marrow is nothing but fat. It is lipids, okay? It's there for cushion for us, all right? So it's composed of vascular and the endosteal compartments, all right? So if you think back to the video, he was talking about when he first stuck that needle in to deaden the bone and to deaden the skin around it. He talked about going into the peristeum, okay? The peristeum is actually a cover over the bone, and you kind of got to go into it to get the bone or to get the deadening to the tissue, to the bone itself, okay? That way they don't feel it. All right, so I'm going to be gross for a minute. You guys ever ate ribs? I like ribs, okay? Are you a bone gnawer? Or are you just one of them that leaves a little meat on the bone after you eat it? See, I'm a bone gnawer. I'm like, pull it down to the bone, like the dry bone after I get through. If you've ever seen anybody eat ribs and then they pull that membrane off while they're eating it, and you get that dry bone, that is the peristeum that they draw off. Does that make sense? Okay? Your bones have that, all right? And so that's what they have to get through to start with to add their anesthesia to it, or the anesthetist material. Lidocaine is all it is. Okay? All right? Then you have vascular compartments, and then the bone marrow arteries and veins. We're going to talk about these in just a minute. And then you have what's called stromal cells, and these stromal cells provide support for us And then you have your hematopoietic cells, which are like your HSCs that we talked about. And then, of course, the immature forms of red blood cells, white blood cells, and platelets. Okay? So these are all there. All right? So this is those trabeculae. I know my drawing wasn't real good, but you got the points. Okay? So the open areas are known as the trabeculae. Okay? So this is where bone marrow is going to be. And you notice it's in the heads of the long bones. All right? As we are young, when we're babies and up to about 12 years old, all of our marrow is red in our bones. Every bone. So you could actually draw marrow out of any long bone on a kid. Okay? They still use the iliac crest most of the time for babies also, or kids. But in theory, you could get it from any bone. Okay? You ever had a baby that, how many of y'all got kids? Any of y'all got kids? Okay, I know some of y'all do. Sometimes babies have a hard time with starting an IV on a baby. You know what I'm talking about? So one thing that we can do, or you can do in a hospital, is they can do an osseous IV. And it basically, they drill it straight into their bone. The reason they're able to do that is because little kids and babies, you can do it in an adult, you just have to do it in certain spots. But babies, they do it a lot of times because all of their bones have red marrow. And so therefore, you get it in that bone and it's eventually going to get into their bloodstream just like anything else because of those veins and arteries that are coming in and out of the brain. What?
Is it as fast as?
It's faster.
Faster.
Yeah. Yeah.
Cool.
Yeah. Especially inserting it, you know, because they do it with a drill, literally, with a drill. They just drill straight into the bone, and then it has a catheter, it goes into it, and they just hook the IV up to it. So, yeah, I mean, it goes in just as fast as it would your veins.
How do they know not to hit the nerve?
Well, the nerves, there's just not that many nerves.
Okay.
There's a few of them, but they're not that many of them to really worry about.
I reckon they have to do that.
Yeah. Okay. Questions about that, guys? All right. Okay. So the endosteel compartment is where basically where we're going to have and we're going to kind of concentrate our discussion. Okay. Side of bone remodeling. Okay. So as we grow, our bone degenerates. It basically gets rubbed off inside our insider bones and our endosteel or endosteum that we have. And so we have these special cells that play a role in remodeling our bone. One of them is known as an osteoblast, and one is known as an osteoclast. Okay? Osteoclast's job is to break bone down. So if bone gets broken off, osteoclast basically engulf it and dissolve it. Okay? The calcium. Osteoblast, anytime you see blast, it's always an immature cell. So it is the one that's responsible for making new bone. Okay? And these guys hang out really, really close to the surface of the endosteel. Okay? So they're in the bone marrow, and sometimes you can actually see them in bone marrow. You can actually see osteoblast and osteoclast in there. But most of the time, they're basically right up against the endosteel, so you don't ever really get them from contamination. Okay? All right? And then, of course, it also contains the hematopoietic stem cells that we talked about a little earlier. All right? Questions about that? All right? So in our bone marrow, we have areas. Okay?
Olives.
Hmm? Little olives? Where do you see freaking olives? Oh, right here. I got you. Your pimento's blue, though, right? Yeah, blueberry in your olive. Should have a pimento pepper. Isn't that what they usually put in olives?
Put peppers in all that.
All right, so I don't know if I've ever told you guys, but several years ago we went to Israel. Me and my wife and my boys, we went. to Israel for a trip, for vacation. Spent 9 days, I guess it was, in Israel, those areas doing all the stuff. So my little one at that time, Avery, he's the 16-year-old non-functioning kid now. He was about 8, I guess. He was about 7 or 8. And they eat different in Israel. You know, I wasn't quite ready for that, you know. So morning breakfast. All right, so here's what Joseph eats for breakfast. Bacon, of course you don't get. No pigs, no pork in Israel. Jews don't eat pork, right? Y'all know that, right? Everybody knew that? Okay, so no pork, so no bacon, no sausage, no ham. Guess what? That's the 3 meats Joseph eats for breakfast, right? So none of that. So you know what their protein was? Pickled fish.
Ooh,
So like pickled herring was what they ate for breakfast.
was it
Okay.
good?
Well, yeah, it was pickled. I mean, wasn't like fried. They didn't fry it for me. It was just there, kind of like sardines, I guess.
Not like-
you know.
Not like
Um,
veal. It's-
Eggs. They did have eggs. They had scrambled eggs. You can get scrambled eggs, but you can't put cheese in scrambled eggs because in Jewish culture, no dairy on meat. So you can get a hamburger, but you can't get a cheeseburger. You can get a pizza, but no meat on the pizza. So you get cheese pizza or vegetable pizza, which is freaking gross. Okay? So, breakfast is not real good in the Israeli neighborhood, okay? Or at least for the American. Food wasn't real good. And salads. Had salad for breakfast. And lots of hummus. Okay? Hummus. They call it hummus. It is actually pronounced hummus. We call it hummus in America. I was told that. Okay? So anyway, so that was my breakfast. So my 8-year-old little boy likes salads and he likes fruit. Okay? So he gets his little salad and gets his plate and he goes and he puts him a big 2 or 3 spoonfuls of grapes on his plate and then heads back to his seat. But when he gets back to his seat, picks up his grape and bites into it, it was an olive. And he goes to spitting. Now, so now I aggravate him all the time about, I need some grapes. You know, want grapes on your pizza? Yeah. So, but anyway. All right. So, so we're talking about, don't make fun of my drawings. Talking about the long ends, or the ends of our long bones. Okay, so as adults, that's where we have marrow at. So at the heads of the femur, heads of the humerus, the tibia, the tibia and the fibula, the radius and the ulna. Okay, those are our long bones. And then we do have some in our ribs. Ribs are actually long bones. The sternum we talked about, our iliac crest, our vertebrae, and our skull. Okay, those are places. All right, so of course we use the iliac crest most of the time. But we're going to use the heads of the long bone to kind of give us an idea of what's going on. Okay? So, your bones we know are hollow. So, from here down is yellow marrow as adults. Okay? Yellow marrow. Alright? Up here though, actually just kind of like right here, because we actually have more yellow marrow that comes up in here. And then you have just a little bit of area. We're going to make this, because that's kind of how it will be, because you have those trabecula. This is our red marrow. Okay? So this is what little bit here produces all your blood cells in your body. Just that little bit. And of course what's in those other bones we made mention of. So in theory, very little marrow. Okay? Alright? So this is where they're at. Alright? So if we could zoom in on this and kind of look at a spot, you would see something like this. Okay? That would have those trabeculae in it. okay, as you go, so those open spots, right? And then you would have marrow inside these areas, okay, that would have this. That's the reason we're able to take that, that I call it the core, you know, it's got a name, it's tuberculae, but that core, and go in and cut a piece of bone out. And when you look at it under the microscope, that long thing, you're actually seeing those tuberculae, or those tuberculae, or how you want to say it. Okay? That's what we're looking for in the biopsy. Okay? So if we could pull then this even out even further, then you would see like the walls, and then you would see cells in niches. Okay? And that's kind of what that picture is letting you know. Okay? So, right up against the wall of the cells, you would have what are known as the endothelial cells, or those bone cells. Okay? This is where your osteoblast and the osteoclast hang out right in this area here. Okay? Then you would have your stem cells that kind of hang out in this area too. Alright? Then you've got an area that you have lymphocytes or lymphoid that they hang out. Okay? And then of course you got vessels. Alright? So the endothelial cells of the vessels. Okay? So because we're forming them in the bone marrow, but we've got to get them into what? The peripheral blood, right? We got to get out those cells that are being formed eventually into the veins, into our arteries, right? Because that's how they get from one place to the other. So they've got to have a way to get into those areas. So some of the cells are really, really close to this, okay? All right, and then one area here, you have what's called an erythroblastic island. Okay? And these are really cool. Because if we zoom up a little closer to it, we would find that we have a big cell that has a butt nucleus with ground glass cytoplasm. What would it be called? A monocyte when it's in the peripheral blood. If it's not in the peripheral blood, what did I say it's called? Somebody said it back there. Macrophage. Okay? So we have this big macrophage who is also known as a mother cell. And why he's a mother cell is because he is actually producing cytokines that stimulate the growth of red blood cells. Okay? So what happens is we have a ruboblast that are surrounding this cell. Okay? All around it. Alright? And then as it goes out, the cells as they go out, you see these big areas of cells, but as they go out away from the mother cell, the macrophage, they become mature red blood cells. So really what you have is you have immature to mature. Okay? So like all the precursor cells are around the mother cell, but then they're pushing the young, older ones out as they go. Does that make sense to you guys? Okay. So that's what like this erythroblastic island is talking about there. Okay. That makes sense? Yes.
What is this
called mother cell. It's a macrophage. It's a monocyte, essentially. If it had been in the peripheral blood, we'd call it a monocyte. But remember what I told you guys, once they leave the peripheral blood, monocytes only stay about 2 hours or so, a few hours. And then they migrate to tissue. Guess what bone marrow is? It's a tissue. And so once they get in the bone marrow, then these macrophages become, helper cells. They secrete cytokines, and we'll talk more about this in the next chapter, chapter 4. But this is just a little introduction, okay? All right, so we'll talk more in detail of this in the next chapter, okay? All right. Okay, so vascular supply, you have 2 arteries. They're known as the nutrient artery and the peristeal artery, okay? Why are arteries important to us? Why do we have to have arteries? Huh? What kind of blood? Oxygenated. That's the key. That's the reason we have 2 different types of vessels. We have vessels that carry oxygenated blood, and we have vessels that carry deoxygenated blood. Okay? So we need to have 2 supplies of oxygen carrying vessels, which we have arteries, but we only have really 1 vein that carries deoxygenated. Okay? And it does its job. All right? So it's known as a central vein, and it drains right back from the bone marrow. Also, most of the time your cells egress. Okay? Have you ever heard that term, egress? All right? Egress means to move or to leave. Okay? So once your cells become mature and they're ready to go into the peripheral blood, then they have to egress into that vessel. Most of the time it's this central vein that it's going to egress into. Okay? And then get to the rest of the body as we go. Okay? All right. So, nutrient artery, what it tells you, it talks about what it does. Then those sinuses are, y'all know what a sinus is? When we talk about a sinus? Yeah, it's an opening. Yeah, it's an opening. A sinus is just an opening. You know, like when you get a head cold, you talk about my sinuses hurt. If you didn't have sinuses, opening parts in your face and in your head, in your skull, then your head would be too heavy for you to hold up. Did y'all know that? Your neck muscle wouldn't be strong enough to hold up your head because your brain weighs a pretty good bit. So you have open spots that make your head lighter. Does that make sense? So sinuses are just big opening areas in those tissue. Okay. We talked about arterioles. They're just small arteries. So arterioles, capillaries, venules. Remember that from phlebotomy. Okay. So it's where gas exchange occurs. All right. Then of course you have some nerves and these kind of things. So this is not as important as some of the other stuff that we're going to talk about. So, but there are pictures for this you guys can look at. All right. Let's talk about the stroma. So the stroma is bone tissue for marrow tissue, and it's there for support. Okay. It provides a meshwork inside the cells, and then the stromal cells produce cytokines that help regulate hematopoiesis. Okay. We haven't talked about cytokines yet. I've made mention of the term a couple of times. But cytokines are essentially signaling chemicals. Okay? They cause reactions to occur. All right? So in our case, most of them are like growth chemicals to like stimulate growth or maturation or differentiation of the cells. Okay? So in other words, what's going to make that hematopoietic stem cell turn into a red blood cell? Or what's going to make it turn into a neutrophil? Those kind of things. Okay? Those are cytokines. All right? Stroma, the stroma compartment of the bone marrow is composed of macrophages, reticular cells, which are known as fibroblasts, and then, of course, adipose tissue. Okay? Macrophages, 2 major functions: phagocytosis, because we don't ever want an infection to occur in our bone. Okay? And so therefore we have these macrophages. That's all they do, is they phagocytize things. They get rid of old cells, help break down those parts that are not being used. Red blood cells. So remember the very first day of class, I talked about red blood cells, erythrocytes. They don't have a nucleus, right? So the ones that are flowing in your peripheral blood don't have a nucleus. But when they're in the bone marrow, they have a nucleus. So eventually, before they get kicked out of the bone marrow and egress into the vessels, they have to expel that nucleus for them to do their job, okay? But you can't have just a bunch of nuclei floating around in your bone marrow from all those red cells that you have created. So you have macrophages that's their job, is they go in and engulf them and then recycle the nutrients from them, okay? Does that make sense? All right. And then, of course, macrophages that secrete hematopoietic cytokines, like the mother cell that we were talking about over there a little while ago. Okay. Second function, serve for that center of the erythroblastic island. That's what we're talking about. Acid phosphatase, positive. Acid phosphatase is an enzyme. Anytime you see that ASE on the end of a word, it means enzyme. So it's a special protein that does a certain job. Acid phosphatase is just a big class of, it's like a bag of Skittles, okay? And then there are particular colors or particular acid phosphatases that we will talk about later on, okay? All right, this is a macrophage. You see that big old cell in the middle, okay? But look at the cytoplasm of it, guys. What does that look like? Ground glass, doesn't it? Now, it's not quite a butt, but it's still kind of got that butt-looking features, doesn't it? All right? Problem is, when they get into tissue, they get real big. Okay? Monocytes do that. But remember, in the blood, they're already big. They're the biggest cell that we have in our peripheral blood. So they just get a little bigger and they kind of spread out. That way, they've got more places to put debris in their cytoplasm. Does that make sense? But those are macrophages, that one big one in the middle, okay? It's actually probably two that's formed together because you've got two nuclei, all right? And they can do that sometimes, all right? Reticular cells, they're located on the abluminal surface. What's abluminal surface mean? Abluminal means what? Below the lumen is essentially what it means, okay? So like a lumen is the hole. So like the hole of the vessel itself. And these basically, they send cytoplasmic processes into the stroma to kind of help with structure. Reticular cells are for structure. They're kind of fibrous cells, okay? Source of CXCL12, which is a type of cytokine that helps maintain our HSC pool in the bone marrow. So it's very, very important to us. This is a very, very important cytokine that's referred to. And usually they're in capital letters and they usually have a number with them. So I'll always kind of point them out to you. I don't necessarily feel like you have to know that, but just FYI, okay? All right, these are what reticular cells look like. And you can see that they're basically forming the structure. You can see they got these fibrous looking projections off of them. And this is what they look like. This is an HE stain. This is used, HE stains are used a lot in histology. So you may not ever come in contact with these. I think it's Hansel-Eason stain is what the H&E stands for. But it's used a lot in like pathology and biopsies and stuff. It's just a real common histology stain. Okay. All right. Y'all know what adipocytes are. Adipocytes are adipose tissue. They're cell that's got a big vacuole in them that's mostly lipids in them. And they provide cushion. They provide support, those kind of things. And they can also help us to provide some steroids and cytokines to influence things. Okay. First 4 years of life, nearly all the marrow cavity is hematopoietic. That's what we were talking about before, okay? So most of the bones, most of the cavities of the bones are red marrow Okay, so they're producing cells. But then after about 4 years of age, they start reducing the amount of red and replacing it with yellow, okay, which is that adipose tissue. And then by the time they're about 12 to 15 years old, they have very little red marrow except in the places that we talked about, okay? Questions about that, guys? All right. And these are just some adipose or adipocytes that are in the bone marrow, in the stroma. Okay? Questions about any of that, guys? All right. Where are we at? What time wise? All right. We'll stop right there, guys. So we'll talk about osteoblast and osteoclasts on Monday. So I will see you guys. Yeah, that's right. Y'all out Monday. So we'll talk about it on Wednesday then. We will have lab Tuesday, so don't forget. All right. We'll also have lab tomorrow, but we will have lab Tuesday. So since we're not having a lecture, so I will see you guys then. Have a good rest of your day.
When we do are we doing the little practical
Yes.
tomorrow?
Yes.
Is it just these ones, or is it going to be?
No,
like
it'll be just
macrophages?
these ones.
Okay.
I never heard that term. These ones.
Yeah.
Yes, it will be these. You just say these. It will be these. You don't have to put the ones in it. These. That's that Yankee talk.
And she from
You don't